So we've been doing some research on the neuropathology of COVID in the last couple of months. And as you know, the next slide, I can do it myself. Okay.
And you go onto the presentation mode.
Okay,
Yep,
now it's better. So you know that COVID-19 presents with a wide variety of neurological symptoms, and I've listed them here. And you know that the most common ones are loss of smell and a loss of taste, but there are also some more severe ones, which I've listed here, and then don't want to mention them now. You all know them better than I do. And also that, I mean, COVID popped up, and then it opened some really, really important questions about how these symptoms of the brain relate to the virus. And of course, we were asking ourselves, so can the virus SARS-CoV-2 be found in the CNS? And then if yes, then how does this virus gain access to the CNS? And as you know, there are like two main routes. So it could either be via brain nerves or nerval systems in the brain, olfactory system or the vagal system that innervates the lung, or it could be via transit of the virus through the blood-brain barrier crossing via endothelial transport. And of course, once the virus is there, the question is, how does this virus cause the symptoms in the brain? Is it somehow really a virus-induced effect, or is it an indirect induced effect? And we believe that neuropathology can really, really be of great importance in these studies, as it bridges like the last gap, looking at patients that died of COVID. And in Hamburg, in Germany, we're very lucky because we're like a city state, so it's an own like a governmental infrastructure. And from the first onset of COVID, our government decided that they would pull a law which exists in Germany, which makes autopsies in extraordinary situations like COVID and pandemics obligatory. That means that from the first patient we had in Hamburg until about the 280th patient listed down here in Hamburg, we autopsied every single individual that died from COVID. And that means that in neuropathology or in pathology, we not only got the patients that were sent or were done by us in pathology, dying in hospitals, but we also got in patients that were diseased and were filtered out at the crematorium. We also got non-natural deaths, and also, as I mentioned before, these autopsies that were ordered by authorities. That meant that very early on we had a fairly large sample number and could really look into the neuropathology of COVID in great detail. And we've done this, and I think this brought us several advantages. One of them is that what we actually looked at in the neuropathology represents also what's happening in reality. We had about 20% of all deaths coming from patients that died at home, 20% died at retirement homes, and 60% died at hospitals. And this means we can now do subgroup analysis, looking at disease groups. We were able to do evidence-based data on mortality rates. We could look at this organ-specific workup, which we do the neuropathology of. And also very important for us, it meant that autopsy has finally gotten some kind of public acknowledgement. I mean, we were in the media, and actually the people on the street realized that autopsy is of benefit, directly benefit to their health. And this gives you an overview of all the 258 patients that we looked in more detail, that we autopsied, and you see that the median age we have is 83 years. So it's pretty old median age at death. And this meant that very early on, as early as May last year, we were able to say that SARS-CoV-2 is actually a multi-organ virus. It affects not only the lung, and this is just looking at viral copy numbers in lung and in different organs. And you see that not only did you find virus in the lungs, but also you find them in the brain. And in the brain, you see that there's a lot less happening than in other organ systems. But that was important because it meant that finally, people were aware of that this is a multi-organ, a virus with multi-organ tropism. And so we looked at the neuropathology in great detail. And I mean, I give you like the main findings up front, and I will go into them in more detail in my talk now. So what we saw is that, and this data is now based on about 110 brain autopsies we looked in a standardized, detailed fashion. And we think that the underlying neurological disease are really likely to influence the neuropathology of COVID-19. So if there's like a pre-existing dementia, this greatly influences how the neuropathology of COVID looks. And also that also if you look into the immune reaction, we did not see clear-cut encephalitis, cerebral bleedings, and no acute necrotizing lesions. And this I'll show you in Uppsala. Let me go back. Let me go back. So this, so we did not find, and as you also realized that what we also saw is that very early on in the pandemic, there were about 1/4 of all patients died of thrombotic incidents, and we also saw these in the brain. But this has ceased now due to the therapy that is based on inhibiting these thrombosis. And if you go in more detail on the neuropathology, what we've seen a perivascular neuroinflammatory response, which is characterized by the activation of microglia and infiltration of cytotoxic T lymphocytes, most pronounced in the brainstem and in the cerebellum. And I've shown you this in this slide. This slide just shows you stainings for IBA1 is a marker for microglia. CD68 is a marker for microglia that are able to phagocytose, and TMEM119 is a specific homeostasis marker for microglia. And you see that we can find different signatures in the brain. We see one signature is that we see the appearance of these microglial nodules, which are characterized by activated microglia and CD8 T cell help. And then around the vessels, we saw a really pronounced microglial activation, which is characterized by this dense infiltrate and dense activation of astrocytes and microglia around the vessels. And then in perivascular spaces, which are localized at the rim of the brain, this was not so pronounced. And there we saw that a loss of TMEM119 of this homeostatic microglia marker. And in the meninges, we did not see signs of manifest, really massive viral meningitis. This was rather a mild response. And when I say we saw this kind of perivascular activation of microglia and also astrocytes, this is really very nicely visible in this one individual, which I picked out here. This is the youngest person that at least I've seen in our cohort, is a 29-year-old male, completely healthy. He had a fulminant course of COVID-19. So only 2 days before he died, he had first symptoms of fever and coughing. And then 1 date prior to death, he had high fever, was found unconscious in bed by partners where he lived. They tried cardiopulmonary resuscitation, but it was unsuccessful. So he died at home. And in the autopsy, it was really amazing because this person only had the typical lung processes that you find in COVID-19, but all the other organ systems were without really findings. So what is astonishing in this patient is that when I say we have this perivascular response, you see that really around all vessels, and this is in the frontal cortex here, you have this pronounced, just have to go back, I'm sorry. accidentally skipped a slide or something. So you see here, this astrocytes that are lined around the blood vessel, and they seem to, also increase the size of their footprints at the blood-brain barrier. And there's also a pronounced microglial activation around this vessel, which you see here. So this is what I mean when I say this kind of perivascular immune response. And of course, we also tried to look into what are the underlying processes that occur. And we did proteomics in these brains, not of all. We only did it of about 10 brains and 10 controls. And what we found here is that if we did pathway analysis, we found a massive induction of the IL-6 pathway. And as you know from the lung pathology, this is also like the most prominent inflammatory pathway that is stimulated in the lung. And it seems to be that in the brain, at least in the ones we looked at, this is also one of the key pathways that is induced. So the next thing we looked into was the question, so where can we find the virus? And I'm telling you that we found the virus in about 50% of all patients we looked into. And I'm also stating here that the correlation between the neuropathological findings we had and the amount of SARS-CoV-2 was not strict, it was not really there. And if we looked in more detail, so where can we find the virus? So we find the virus in immune cells in the brainstem, but also in cranial brain nerves and in endothelia. And I will show you pictures of this just now. So this is when I say that in about 50% of patients we found SARS-CoV-2, this is what the data looks like. So this is, a very colorful slide. Don't get confused. This is only a slide showing the first 43 patients we looked at, and we also reported. And then they're sorted by amount of virus found in the brain. And you see that, this darker, colored squares here, is where we found virus. And the more to the right, the more virus we found. And this kind of red and blue and yellow dots just represent individual patients, different brain regions, and the amount of changes we found. And you see, and red means a lot of changes, and blue means almost normal. And you see that there's no strict correlation between the amount of virus, which would be on the right side, and the changes we see in the brain. This is what I meant when I said there's no strict correlation between amount of virus and neuropathological changes seen in these brains. And let me just go the next slide. So where do we find the virus? I told you that we find the virus in cranial nerves originating from the brainstem. So this is one example, or actually two examples of viral. So this is viral spike protein found in the vagal nerve. So this here would be all vagal nerve, which here is entering medulla oblongata, and here would be the vagal nucleus. So this is at least one hint to us that maybe the vagal nerve might be involved in neuroinvasion. And I told you that we find the virus also in brain parenchyma, in immune cells in the brain parenchyma, and here in brown, again, this is a virus stain. And here in a fluorescence slide, you can see that in green we have the viral spike protein, and in white astrocytes, and in red activated microglia. And we think that the virus is here contained in an activated microglia cell in the brainstem that might have phagocytosed the virus. And finally, I told you that we find the virus also in vessels, in endothelia. And here's an example of brain endothelia. 2 patients where we found the virus. It's very staining in the very, lumen-most fraction of the endothelia. Here, this brown staining would be, again, virus. And here, again, this is virus. This is an electron-microscopical picture where but colleagues looked for a long time to finally be able to find virus. And this is actually, I think, this SARS-CoV-2 coronavirus, which is found perivascular, again, in the endothelia. So maybe, transfers from the endothelia in the brain. So at least in Germany, there's now a big change of the virus subtype. So we're moving from the original strain that we have to a strain which is this B1.1.7, which at least in Germany, it's called British SARS-CoV-2 strain. I don't know what it's called in the UK. But in this SARS-CoV-2 strain, people were always asking us, so how does the neuropathology look like? And I can tell you that the neuropathology looks just as it does in the other corona patients we looked at. So there's no differential, neuropathology in this corona strain. In this, this is a patient that was 70-year-old, so an older female patient that again had a fulminant course of the disease and died at home and was found unconscious by her husband. And then again, they were not able to resuscitate her. And again, this is one of the patients that we only got because of this kind of autopsy scheme we implemented. So interesting in this patient is the fact that we also looked at viral content all over this patient. And you see that the highest content of virus, of course, was found in the respiratory system. But here, the second highest content of virus we found in the olfactory mucosa. So, like maybe olfactory neuroinvasion, but also in the vagal nerve. So the vagal nerve has a higher titer than the others, and also you see here in the carotid artery. So again, this vascular kind of pronouncement is also seen here. So I hoped that with this kind of excursion into neuropathology, I could convince you that neuropathology can really contribute to clearing up at least some of the features associated with COVID-19. And so if you would like a summary from me, I would tell you that yes, we can find SARS-CoV-2 in the brain frequently. More than half the patients have it. And I think that SARS-CoV-2 gains access to the brain both by the olfactory system, the vagal system, and by blood-brain barrier crossing. Maybe differentially in different patients, but in individual patients, we found, we have findings that speak in favor of these three systems actually being active. And I think that's also the notion that is followed by most researchers now, is that an overacting immune system is the thing that damages the brain. It is not virus-induced direct effects to the brain. And with this, I'd like to finish this talk. I hope I kept my time. And I want to thank the people that were involved. And you can see the names here. And of course, this was a close collaboration between neuropathology, pathology, forensic medicine, which brought us all these kind of non-hospital cases, which we can study now. And also people from microbiology and virology were involved in this study. So with this, I would like to... Yeah.
Thanks so much, Markus. What a fantastic talk, and particularly the data on the recent, as you called it, British variant. I mean, we call it the Kent variant, or there are other names for it. So we've got a few questions now. We're running a little short on time, and we have to strictly end at 8 o'clock. So just if you can very briefly say, did you find in the perivascular microglia, could they be macrophages? And did you find anything in the spinal cord? Those are the top two questions. And I would say thank you to others for answering questions. Perhaps you can answer those in the chat during the session or after
I see in the chat. And so, yes, I think that, I mean, some of them are actually macrophages that, come from the blood system that, invade the brain. And I mean, where there are markers of all of these. And I think it's a mixture of microglia and macrophages. Yeah, that's the first. And the second question was, I forgot.
Are you finding damage in the spinal cord?
Yeah. So I'm sorry, we didn't check spinal cord yet. So we only looked at I think 12 regions in the brain until now. We have all the paraffin blocks, but we didn't do it until now. We're still autopsying a lot of patients now, actually more than in the beginning, and we're just working up these also as they come along.
So thanks very much. I mean, there have been, as of today, on the Johns Hopkins dashboard, 115.8 million cases of COVID, more than 2.5 million deaths globally. And one question I saw was, what is the relevance of this to long COVID, particularly with the vagus nerve and some of the symptoms that people with long COVID have, currently estimated at 10% perhaps of people, which could be 12 million. But do you have any insights into that?
Well, what we have right now is, so we don't know, because I mean the patients that die of COVID, as you know, is only about 1.1 or 2% of all patients that have COVID die from it. So we cannot say anything about patients that survive. We only see the really severe ones, no? But what we have now is we have some patients that had COVID half a year ago and that now died half a year after they have had COVID from something else, no? And we're looking into these now in great detail, because these are the ones that will tell us, how does the brain, I don't know, how does it cope with COVID once, it's over, Yeah. We haven't looked into this in great detail, but we're doing it right now. This is, I think, a very important cohort.
Okay, thanks again so much.